Hello friends, in last section I've talked about I was talking about the epithelium, and there I have discussed about the types of the epithelium, the classification of the epithelium, and the locations of the various kinds of the epithelium, that where are those epithelium located, for example cuboidal or columnar or squamous epithelium. Further, I've discussed about the functions of the epithelium, and in this video, in this section, I will be talking about the details of the epithelium, that what structures are present over the apical surface of the epithelium, and what structures are present in the lateral surface of the epithelium and the basal surface of the epithelium. So now let's try to understand this concept with the help of diagrams. So here I'm taking the example of the columnar epithelium, because on the apical surface of the columnar epithelium there are the cilia, because they are present, especially the pseudo-stratified columnar epithelium. They are present, I have told you that they are present in the respiratory passages, for example in the trachea. There, with the help of the cilia, these kind of epithelium helps to get rid of the mucus or the secretion that are secreted by the goblet cells. Okay, so here I'm trying to draw the diagram of the columnar epithelium, just one columnar epithelium, so that I can show you the surfaces and I can show you the structures present over it. So here, actually I should draw it little downwards so that I can show you the cilia also. So for example, here is our basement membrane, okay, and here is one epithelial, actually columnar epithelium. Okay, so this is one example of the this is one cell, for example, columnar epithelium, and remember the epithelium is avascular. Epithelium is made up of the closely tight cells that adhere to each other. For example, here also there are the epithelial cells, and there it is spreading over it. So epithelium, remember epithelium, they are made up of the closely tight other cells. They are closely attached to each other, very strongly binded to each other, and epithelium is avascular, which means they don't have any kind of blood supply. So from where they are getting the nutrition, as they are avascular, so they are getting their nutrition from the basement membrane. We will talk about the basement membrane, what are the structures present in it. So first, what I will discuss in this video, that I will talk about the structures present over here. This is the apical surface of the epithelium, this is the lateral surface of the epithelium, and this is the basal surface where the basement membrane is present. So let me talk about first the apical surface, what structures are present over here. Then we will talk about the lateral and basal surface. So for example, in the apical surface, this is our apical surface. This is our lateral surface, and this is our basal surface. Basal surface. Okay. So in the apical surface, what structures are present? This I have drawn here. This is the microvilli, somewhat bigger. This is the stereocilia, and this one is the cilia. Remember, so let me make those clear, that sorry, this structure is the microvilli, this is stereocilia, and this one we call this as cilia. Now remember that microvilli and stereocilia, they are made up of the actin filaments. So a lot of actin are present over there. Okay, so for example here I am drawn with this A. A shows the actin, for example. So they are made up of the actin filaments, various actin filaments, and but they are not able to move themselves. They are immotile. They are non-motile. They are not mobile at all. So microvilli and stereocilia, they are not mobile, means they can't move. But this is structure, ciliary portion of the epithelium or the cilia of the epithelium, it can move. This is motile. It can move himself. So over the surface, apical surface of the epithelium, there are three kinds of structures over there: microvilli, stereocilia, and cilia. Among which microvilli and stereocilia are non-motile; they can't move themselves, and the cilia can move himself. And cilia is the motile structure. What is the function of the cilia? Now cilia helps to get rid of the mucus secreted by the goblet cells in the respiratory passages. So I have mentioned about this, about the immotile cilia syndrome. I mentioned about this in the previous section also, that some unfortunate patients have the genetic defect of the ciliary protein. They don't have much developed ciliary proteins over their epithelium, over the apical surface of their epithelium. So what it causes, that they are not able to get rid of the mucus secreted by the goblet cells in our respiratory passages, for example in trachea. So if they have the problems in this cilia, because it is moving like this, cilia is moving like this. So for example, this is our trachea, and this pseudostratified columnar epithelium is present. I have taken this as columnar epithelium, for example. So this is our trachea. Suppose this is a trachea, and the ciliary proteins, this cilia is present over here. So what are the functions of this cilia? So they are, what they are doing, they are clearing up the mucus present in the trachea, so that there could be the proper demand of the oxygen, carbon dioxide, proper demand of the gas exchange over there. So that we can inhale or exhale the gases. For example, inhale the oxygen, exhale the carbon dioxide. So if those patients who are suffering from the immotile cilia syndrome, okay, so those patients, what will happen? These cilia will not be able to move properly, because they are not much developed, or they are underdeveloped. So if they will not be able to move properly, so they will not be able to clear up the mucus present in the trachea. So if the mucus, or if the secretions will present over in the trachea, and they will start to accumulate in the trachea, and what will happen? It can cause these secretions or mucus in the trachea for a long time. It is not so good. So it will cause the severe chest infections, various kind of respiratory infections. So these people have the problems, have the complaints of the severe chest infections, severe respiratory infections, and many kind of infections which are related to our respiratory system. So these cilia are very, very important over the apical surface of the epithelium. So if we talk about the details of the cilia, that what really helps these cilia to move, okay, what really helps these cilia to move inside the trachea. For example, if I am talking about the trachea, so what structures helps it to move? So actually, if you talk about this portion, the basal portion of the cilia, the base of the cilia, and about this portion, so I'm talking about this portion right now, that they have the microtubules inside it, and these microtubules are arranged in such a fashion that they have the 9+0 arrangement, 9+0 arrangement of the microtubules. Now what is this 9+0 arrangement? And over here, so in the basal surface, in the base of the cilia, in the base of the cilia, in the base of the cilia, 9 plus 0 arrangement is present over there. But if we talk about the portion over here of the cilia, in the mid portion of the cilia, 9 plus 2 arrangement is there. 9 plus 2 arrangement. Now what does this really mean, that 9 plus 0 and 9 plus 2? Actually, if you will go into the detail of the ciliary portion, ciliary structure of the epithelium, then rather ciliary structure present over the apical surface of the epithelium, then just focus over here, that for example, this is the portion of the cilia. I am magnifying it, and if I am talk about this arrangement, the base of the cilia, that is 9 plus 0 arrangement, they are arranged like this. Actually, there are microtubules. There are microtubules over there. So they are making the triplets over there. They are making the triplets over there. So look, now I have drawn these triplets in the form of 9. So that's why I am calling it 9. Here I have drawn the 9 plus 0 arrangement. So these are the microtubules. What are these? These are the microtubules over here. Microtubules. These are the microtubules over here. And they are bind like this. So they are coming like this. Okay, so these are the radial fibers over here. So what I'm trying to explain, that these microtubules, they are arranged in the circular pattern in the count of 9. So if I am talking about the one magnified portion of the cilia, that they are in the circular portion, these microtubules are arranged in the triplet forms, and they are present in the number 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. I've drawn it 9. These are the triplets, and in the center there is nothing. There is 0. So 9 plus 0 arrangement. That's why I'm calling it the 9 plus 0 arrangement. In the 9 plus 2 arrangement, what will happen? That, so this was the 9 plus 0 arrangement. 9 plus 0 arrangement. They are present in the base portion of the cilia. And if I will talk about this portion, for example, so I am talking about it over here, okay? Here. Okay. So 9 plus 2 arrangement now. 1, 2. They are making the doublets. So they are making the doublets, and they are also present in number of 9. So these are the 9 doublets over here, and they are connected with these two central microtubules. So they are also arranged. They are also arranged with these radial fibers over here. So this is the 9 plus 2 arrangement. This is the 9 plus 2 arrangement over here. So this is the 9 plus 2 arrangement. So what makes them really move cilia? They are moving in the trachea. So what they are really moving? They are not present only in the trachea, because these epithelium, columnar epithelium, are found in other portions too. For example, in the reproductive system in the female. For example, if I will talk about the immotile cilia syndrome, if the female is suffering from the immotile cilia syndrome, they may be sterile. Why? Because the oocyte is not able to move in the uterine tubes, because oocyte is moving with the help of cilia. So oocyte is moving in the uterine tubes with the help of cilia. If cilia are not present over there, or the cilia are underdeveloped, oocyte is not able to move, or oocyte is not able to uh, reach to its destination. Fertilization will not occur. So they could be sterile. At the same time, if immotile cilia syndrome is found in any male, male can also be sterile, as the spermatozoa, as it can interfere with the mobility of the spermatozoa. So again, cilia are present, cilia must be there for the mobility of the spermatozoa in males also. So if the males, some males are suffering from the uh, immotile cilia syndrome, they could also be sterile. So these cilia are more important. Now I've told you that they are arranged in the 9+0 and 9+2 arrangement, and I have explained you about these structures, that what they really means. They are the microtubules. Here they will be in the doublet form, here they will be in the triplet forms, and actually they have two more structures. If you will look at here, these microtubules are bind together. So these microtubules are bind together by the help of which structure? They are bind together with the help of Naxin protein. So Naxin molecule is helping these microtubules to bind with each other. If you will look over the microtubules, what are the structures of microtubules? They are present like this. They are present like tubes. Okay, so they are present like this. These, if I will magnify this one, so they are present in the form of these tubules. So these are the microtubules. They are present like tubes. That's why they called as the microtubules. So Naxin molecules help microtubules to bind with each other, here as well as here. And there is one more molecule which is very important, which helps them in the motility of cilia. So what is this? What is that structure? Actually, dynein. Dynein molecule. What does dynein do? Dynein is responsible for the moving of the cilia. So if the cilia is moving, that means they are taking the help of the dynein molecules. So dynein molecules are responsible. Dynein is responsible for the mobility or the movement of the cilia, and Naxin molecules are present. Naxin is responsible for the binding of the microtubule. So Naxin is responsible for the binding of the microtubules, and dynein is responsible for the bending of the cilia. So this was about the ciliary structures and about the apical surface of the epithelium. Now let's move on to the lateral surface of the epithelium. As I have told you that these epithelium binds to each other very tightly, and they don't allow any substance, unless it is very small or lip of lip. Does not allow, these epithelial cells does not allow any structure or any substance pass through it, at least paracellularly. So for example, here is also one epithelium over here, and these epithelial cells bind so tightly to each other that they make sure that any kind of substance do not pass them or do not penetrate them, at least paracellularly. Paracellularly means between the cells. If you talk about the transcellularly means throughout the cell. So if any substance wants to pass in between the cells, that kind of transport is the paracellularly. So these epithelial cells do not allow any substance to transport paracellularly. So with what structures they bind very tightly to each other? So let's talk about those structures. So if I will, let me draw one epithelial cell over here, and let me show you. So here I have drawn one more epithelium cell, one more columnar epithelium cell rather. This is the nucleus of the cell, and here I am extending the basement membrane. Okay, so let's talk about here. Here are some structures actually, they bind to, which helps them to bind these epithelial cells to one another. So what kind of interaction is it? This we call this as, this interaction is called as zona occludens. Zona or zonula occludens. So this is the zonula occludens. Also, there are more adhesions over here. These are zonula adherents. These are zonular zona adherents. Okay. Also, there are some other structures like you have heard about the desmosomes. They helps in binding of the cells to each other. So desmosomes are also present over here. Desmosomes are present over here, which also helps them. So desmosomes, or they are also called as macula adherents. Macula adherents. So they also helps to bind the cells very tightly to each other, or the epithelium very tightly to each other. They bind the epithelium. So also there are one more structures which allows these epithelium to interact with each other with the help of the, for example, with the help of sodium ions or calcium ions or CAMP. So what are those structures? Those are actually some kind of gap junctions. So these are the gap junctions over here. Gap junctions. Okay. So mainly these four structures are present in between the epithelium, which makes sure that any kind of substance does not pass them through paracellularly. So what are these structures? Zona occludens, zonula adherents, desmosomes or macula adherents, and these gap junctions, which help them to interact with each other, these epithelial cells. Now, if I will talk about the zonula occludens in more details, there are present two kinds of proteins over there. Actually, these proteins are claudins and agglutins. So what kind of proteins are these? Claudins and occludins. So these are two proteins which are present in the zonula occludens. So occludens, name signifies that they have the protein occludins and the claudins. Now, if you talk about the zonula adherents, there are also many kind of proteins over there. For example, E-cathedrins or vanillins or alpha-actinin, which binds to the actin filaments and helps them to give them more strength in binding to the epithelial cells, bind to each other. Also, desmosomes, there are also some kind of proteins over there which helps them to bind to each other, and the gap junctions with the help of some kind of proteins. For example, connexons, actually they are the five, in the circular portion there are the 5 cells, connexins, which makes together connexons. Okay, so these connection proteins helps for the helps these epithelial tissues or epithelial cells to interact with each other with the help of sodium ions or some kind of calcium ions, some CAMP. So This was about the lateral surface, that how they are binding to each other and how they make sure that any kind of substance should not penetrate them in between the cells, at least paracellularly. So I talked about that apical portion of the epithelium. Also, I talked about the apical portion of the epithelium. I talked about the lateral surface of the epithelium, that how they are connect to each other and how they interact actually with each other. Now I'm going to talk about the basal surface. As I mentioned, that epithelium is avascular, they don't have any kind of blood supply over there. So how they are gonna take the nutritions? So they are taking the nutrition from this basement membrane. As these epithelial cells, at last they rest on the basement membrane. If I talk about the simple epithelium, for example, simple squamous, simple columnar, simple cuboidal, they simply rest on the basement membrane. And if I will talk about the stratified, those epithelium which have the multiple layers. So stratified epithelium, for example, stratified squamous, stratified cuboidal, stratified columnar epithelium, they also rest on the basement membrane. But if we look over the surface, or if we look over the top surface of these stratified epithelium, we would get to know that what kind of epithelium is it. But at last they rest on the basement membrane at least. So this basement membrane, in the basal surface of the epithelium, basement membrane is present. They rest on the basement membrane. This basement membrane is helpful in providing some kind of nutritions to the epithelium, as they are avascular. So what kind of structures present in this basement membrane? Actually, in basement membrane, let me rub this portion so that I can tell you about this. So let's talk about the basement membrane on the basal surface. Basement membrane and what structures are present over there. Basement membranes. Okay. Now, actually, in the basement membrane, there are two structures over there, two main structures over there. What kind of structures are these? They are basal lamina, and the zona reticularis. Zona reticularis in this basement membrane. Now first let's talk about the basal lamina, and then we'll talk about the zona reticularis. In the basal lamina, they have further two more structures over there. What kind of structures are these? These are so in the we have in the basement membrane, again in the basement membrane we have the basal lamina, and we have the zona reticularis. So we have in basal lamina we have lighter portion. In the basement membrane we have the lighter portion, and we have the darker portion which is more denser. Okay, so we have zona lucida for the lighter portion and zona densa. Okay, so this is the lighter portion of the basement membrane, this is the denser portion, more compact portion of the basement membrane. Okay, further, and zona reticularis, they have some proteins which helps epithelial cells to get some kind of nutrition from it, and they have some kind of fibers. So let's talk about what kind of fibers and what kind of proteins are present over there. Okay, so if we talk about the zona lucida, so what are present over there? In the zona lucida, we have integrins, we have the laminin, and we have antactin. So these are the three proteins present over there in the lighter portion of the basement membrane. If I will talk about the basement membrane over here, let me show you. This is the basement membrane. Okay. Besides it, underlying it, because the epithelial basement membrane, what is the function of the basement membrane? It separates the epithelium from its underlying tissues. Okay, so if I will magnify this basement membrane of the epithelium, on which the epithelial tissues or epithelial cells rests on. So this is the lighter portion of the basement membrane, and here this is the more denser portion. So this is the more denser portion of the epithelium. This is more denser. So this is, I am calling it the zona lucida, and this one is the zona densa. Zona densa. And underlying it, there is zona reticularis. This is the zona reticularis. Now, in the zona lucida, we have some proteins like integrins, laminin, and entactin. If you talk about the zona densa, again, we have some kind of fibers and some kind of proteins. So there is collagen 4 fibers, and fibronectin. So these two structures are present in the zona densa, which is more compact one. So this is the zona lucida, zona densa, and this is the zona reticularis. This one is the zona reticularis. Okay, and this whole structure is the basement membrane. Okay, now what structures are present in the zona reticularis? We talk about zona reticularis, there are two kinds of fibers present over there. Now these fibers are these fibers are the collagen 7 fibers and collagen 3 fibers. So these two fibers are present over there in the zona reticularis of the basement membrane, which helps these basement membrane structures to separate it from the underlying connective tissues with the epithelium, and also provide some kind of nutrition which epithelium wants. So these are the structures about this basement membrane. So first of all, I told you about the basic structure present over there, basal lamina and zona reticularis. For the basal lamina, have two portions, zona lucida and zona densa. In the zona lucida, we have integrins, laminin, and entactin proteins over there. In the zona densa, we have collagen 4 fibers and fibronectin, which supports the basement membrane. And the zona reticularis have the collagen 7 and collagen 3 fibers. So this was about the basal surface. So in conclusion, what I talked about the structure, what structures did I discuss till now? That I have talked about the apical surface of the epithelium. There I have told you about the microvilli, stereocilia, and cilia are present over the apical surface of the epithelium. Microvilli, stereocilia are non-motile, they can't move, and cilia is the motile. Dannin is helpful for the bending of the cilia or the movement of the cilia. Nexin molecules are responsible for the binding of the microtubules together. In this portion, we have the 9+2 arrangement. In this portion, we have the 9+0 arrangement. Next, we move on to the lateral surface of the epithelium, There I've told you that there are the various kinds of jonula presents over there: jonula occludens, jonula adherens, macula adherens, or the desmosomes, and the gap junctions. So in jonula occludens there are the claudins or occludins proteins present over there. In the jonula adherens there are also some more proteins, for example alfactinin or ecathedrins proteins. So some kind of protein, these proteins helps them to bind with each other very strongly or more prominently. Uh, Further, we have the desmosomes or macula adherens, which also helps to have these epithelial cells to bind to each other, and gap junctions with the help of some connections proteins, uh, and with the help of some sodium ions and calcium ions, some CAMP, so that they can interact these epithelial cells to each other. Further, on the basal surface, we have the basement membrane, which separates epithelium from the underlying tissues or underlying connective tissues. In the basal surface, in the basement membrane, we have the basal lamina and zona reticularis, two main structures. If you talk about the structure, here I have drawn it. This is the basement membrane. The first one is the zona lucida, the more lighter portion. Zona densa is the more darker, more compact one. And underlying it, there is the zona reticularis, and here will be the connective tissues. And above it, there will be the epithelium. Above it, there will be the epithelium. Okay. Basal lamina have zona lucida and zona densa. Zona lucida have three kinds of protein: integrins, laminin, and actin. Zona densa have collagen 4 fibers and fibronectin, and zona reticularis have the collagen 7 and 3 fibers. So I hope this would be helpful for you to understand the epithelium. This was all about the epithelium, some basic histology of epithelium. So thanks for watching this video. See you in my next section. Thank you.